Hello, my name is Romina Zambrana Montaño, and I'm going to talk about evolution of SARS-CoV-2 Gamma variant during the second wave of the COVID-19 pandemic in Argentina. Throughout the pandemic, Argentina has been characterized by 4 waves of infection until now. While the third wave associated with Omicron was characterized by a huge number of infections in a short time, the second wave associated with the Gamma variant triggered not only a large number of infections in a longer period of time, but also caused the highest number of deaths recorded in the entire pandemic in the country, in a context where vaccination had just started. This wave was associated with the introduction and rapid spread of the variants of concern, including the variant named Gamma. The Gamma variant, despite being highly transmissible and having a high incidence in Latin America, his evolution has not been studied with the same depth as other variants such as Delta or Omicron, with a broader distribution in different regions of the world. Data we used was Argentinian SARS-CoV-2 whole genome sequences belonging to Gamma variant and its sublineages, downloaded from GISAID database. Here you can see the number of samples taken from each province. These sequences corresponded to individuals diagnosed from February to December 2021, obtained with Oxford, Nanopore or Illumina platforms, in the context of the molecular surveillance implemented by PAIS project. We also extracted 5 SARS-CoV-2 sequences with a high similarity for each query sequence from the GISAID database using the Audacity instant tool. Later, alignments were built with the MAF program using default parameters. With the aligned sequences, phylogenetic analysis was performed by the maximum likelihood method with IQ3 software to identify introduction events and to study their diversification process. From the phylogenetic analysis, we found that the lineage P1 was predominant throughout the second wave in Argentina, as well as P115 and P12 sublineages, followed by lineages P114, P117, and P113 to a lesser extent. The phylogenetic analysis shows several introductions of the Gamma variant to the country. They are represented by pink arrows in this figure. Next to the tree we can see 2 clades as an example where we can observe that the Argentinian sequences not only form monophyletic groups between them, but also with sequences from other countries such as Brazil, Paraguay, and the USA. Small gamma clades that contain sequences from a specific region suggest a more regionalized local transmission in some cases, like the one we can see on this map, where the different provinces are differentiated by colors. But the large group of lineage P1 that contain most of the Argentinian sequences from north to south of the country show a wide local transmission and diversification of common transmission change. In the case of sublineage P115, we observe the same transmission pattern as in lineage P1, where sequences belonging to both the north and the south of Argentina are grouped. Within the large clade, we can also analyze in more detail how the infections were introduced. For example, we can observe an introduction to the province of Rio Negro, which was extended to the continuous province, which in this case is Neuquén. For other monophyletic groups, it is more complicated to define in detail the transmission change. However, we can obtain information on the provinces in which the lineage P115 circulate in. We can even suggest that there was some transmission events shared between Argentina and Chile. For sublineage P12, cluster included sequences for a few Argentinian provinces, which suggest a more limited geographical distribution. In this case, we are seeing the largest clade that allow us to appreciate an introduction to the province of Tierra del Fuego and a subsequent diversification that apparently reached other provinces such as Santa Fe and Buenos Aires. In summary, as in other South America countries, Gamma was the main variant in the second wave of the COVID-19 in Argentina, originated from several introductions and showing a wide distribution with circumscribed and extending transmission change throughout the country. These results reveal the impact of the regional circulation of SARS-CoV-2 variants on the epidemiology of Argentina in the second wave.